Hey everyone, and welcome back. So in this video, I'm going to go through the different types of dominance that we see in nature. We'll talk about polygenic traits, and we'll go through how to do a Punnett square. We'll talk about monohybrid and dihybrid crosses, so I can kind of walk you through that exercise as well. So if you recall from my video on Mendel's laws, one of the things that Mendel talked about is in his concept of inheritance, there were different versions of genes, and we call those different versions of genes alleles or alleles, however you want to pronounce it. Either way is actually fine. But these alleles are different versions of a gene that encode information that will control the outward appearance of that particular characteristic. So, of course, Mendel wouldn't have referred to these as genes. He would call them inheritance units. He didn't have a concept of a gene, but we'll use the modern terminology throughout this video. So in his eyes, when you had two different versions of a particular gene, one of those had to dominate over the other, which means one of them is going to control the outward appearance or the phenotype of that particular characteristic. We refer to this particular model as complete dominance, and the majority of the traits that he examined in his pea plants and he studied for his research were genes that had alleles that had at least one that was completely dominant over the other. We now know that there are different types of dominance in nature. In addition to complete dominance, we also have incomplete dominance, codominance, and then we also have situations where sometimes multiple alleles are involved, and the way those alleles interact produces very distinct and different phenotypic expression. So, for example, with complete dominance, when you have individuals who are heterozygous for a given trait, the dominant allele will completely mask the phenotype. It will control the phenotype. So here you can see you have, this says a purple flower plant. It appears a little more pink to me. But anyways, you've got this purple flowering plant, this white flowering plant. When you combine them together, all of the offspring in the F1 generation, that first generation, are all going to be purple. And the reason why is the purple allele is dominant over the white recessive allele. However, we don't lose that information because when we allow these F1 progeny, these F1 offspring to reproduce, we can recapitulate that white phenotype when we end up with an organism or an individual that is homozygous for that recessive allele. In incomplete dominance, we run into an entirely different situation. Note a couple of things. First off, instead of writing something like big P, little p, to distinguish between a dominant and a recessive allele, notice that we're now using the capital C. In this case, it'll stand for color or the color of the flower petals. And then we write the allele information as a superscript. So CR, CR versus CW, CW. So here in the parental generation, just as we had in over here in this example, we have a homozygous red individual being crossed with a homozygous white individual. But in the F1 generation, we don't see one of the alleles dominating over the other. Instead, what we see is both of these have sort of combined into what we would refer to as an intermediate phenotype. So notice that pink is halfway between red and white. But again, when we do the F2 generation, when we do that F1 cross, get the F2 generation, we will see we now recapitulate all of the different genotypes there. So again, we have our homozygous red, we have our homozygous white, and then the heterozygotes stay pink in that case. So that's one of the ways we can distinguish between complete dominance and incomplete dominance. What happens when you have a heterozygote? If you have a heterozygote and you only have one of the phenotypes, that likely means you're looking at a situation of complete dominance. When you end up with an intermediate phenotype in a heterozygote, you are looking at incomplete dominance. We also have an example of codominance. Perhaps the best known example of this is the human ABO blood grouping. So in humans, whether you know this or not, there are three different conditions essentially you can have with your bloods. I guess four different conditions depending on how you put on it, but there are three alleles out there. First off, you have the little I allele. All right, this corresponds to what we refer to as the type O blood type. Notice that if you have both little I, you are homozygous and you end up with type O blood. However, now when we look at the A and B phenotypes, what you see is now it's big I A or big I superscript B. So what this should be telling you is we're not in the same situation at this point because we have a big I. So what that's telling you is that the A and the B alleles are dominant over little I. And indeed, we find that's the case. So whether you are homozygous for the A allele or heterozygous for the A allele and the little I O allele, you end up with type A blood. The same thing with B. Whether you're IB or IB little a, you have type B blood. If you have type A, if you have one type A gene or one type B gene, and it doesn't matter what the other one says. If it also says A or it says O, or if it also says B and it says O, you're going to end up with the A or the B phenotype. So when we compare the big IA or big IB to little I, that is a situation where we have complete dominance. But look what happens when we have the IA-IB heterozygote. We don't end up with either of the two previous situations. We don't end up with A dominating over B or B dominating over A, nor do we have like a weird intermediate phenotype where we'd have like type C blood or something like that. No. We get AB blood. We have blood cells that express both the A and the B antigen. And understanding how this works goes back to understanding what these particular genes are encoding. And what they are doing is, it turns out that this particular gene controls what antigens are expressed on the surface of our red blood cells. So if you have an A allele, you produce the A antigen. If you have a B allele, you produce the B antigen. And if you have the little I allele, you produce no antigen whatsoever, which explains why just having one copy of the A or B allele leads to an A or a B phenotype predominating over O. O is essentially the absence of any antigens. Whereas having a single gene that encodes A is going to give you the A antigen on all your red blood cells, and having a single B antigen is going to give you B antigens all over the surface of your red blood cells. But if you get both, you don't get some weird combination. No, you get the A and the B antigen on your surface. And this is a classic example of codominance. So the ABO blood group is a great example of codominance in the fact that you don't get an intermediate phenotype. You don't get one completely dominating over the other. Instead, you see both of the phenotypes expressed in their full glory at the same time. Then if we come over here to the multiple allele situation, it gets a little confusing because what you end up in is a situation where you can see now you have the big C brown fur type. All right. So any big C is going to give you a brown money. But then you have the chinchilla, the little CCH, the little CH for Himalayan, and the little C albino phenotype. The big C dominates over all of the other alleles. So one big C is giving you a brown rabbit. But when you get to these weird combinations of different genes, how they interact is gonna lead to different phenotypes because these are all recessive to big C, but they interact with each other in different ways, and you end up with multiple alleles, which gives you lots of different fur colors and things like that. All right. So this is a situation where it's not as straightforward as there's two alleles and they're both competing. No, in this case, there are four different potential alleles that you can have, and the way they interact is going to control the fur color of the rabbit in this case. So let's look at some examples in humans of things that would be considered completely dominant, and I'll talk about the fact that there's actually a little bit of clarity we need to make on some of these. So again, complete dominance is pretty straightforward. If you have two versions of the gene, one of them is going to control the phenotype and the other one's going to pretend like it doesn't exist. One's dominant, one's recessive. For the most part, in humans, when we look at dominant versus recessive genes, cleft chins, the cleft chin is the dominant gene. One allele gives you a cleft chin. If you don't have any cleft in your chin, you are homozygous for the recessive allele. Widow's peak, that little point where the hair comes to, that is a dominant version of that gene. Dimples are a dominant allele. If you have dimples, you have at least one dominant gene one dominant allele for that dimple gene. Brown hair and black hair are completely dominant over blonde hair. So if you have a black hair gene and a blonde hair gene, you're going to have black hair. Okay. Freckles are a dominant gene. So if you have one allele that says you're going to have freckles, you're going to have freckles. Brown eyes are dominant over blue eyes. Now, one of the things I do need to understand, in previous videos, I've discussed the fact that eye color is not a single gene phenotype. That is true. But one of those genes involved in that, in that multiple gene inheritance, one of them controls like brown versus blue. All right. That brown-eyed allele will dominate over that gray or blue-eye allele. Those other genes kind of refine what version of brown eye you have or what version of blue eyes you have. So just be careful how you interpret that. It's kind of a bit like with the ABO blood grouping where you've got a little bit of dominance and then some other stuff going on there. And then free versus attached earlobes is another great example, with the free earlobe being the dominant gene. So if you see somebody whose earlobes are completely attached, you know that they're homozygous for the recessive allele for that particular trait. Here are some examples of incomplete dominance in humans. Incomplete dominance, again, is the appearance of an intermediate phenotype where one allele cannot dominate completely over the other, and the heterozygotes show that intermediate phenotype. So, one example here is for curly hair. If you are homozygous for the curly hair trait. Now, I don't know why these are written big C and little c, because that's not the way it's supposed to be. This is a graphic that doesn't have the same labeling, but what I should be seeing is something like big C, little c, big C, superscript S for straight, right? Superscript C for curly. If you have two of the curly genes, you end up with curly hair. If you have two of the straight hair genes, you end up with straight hair. And if you end up with one of each, you get the wavy hair phenotype. So if you have perfectly straight hair, you have the recessive, you're homozygous recessive for that. That's me. I have like the straightest hair imaginable. If you have a little bit of a wave to it, you're a heterozygote for that particular gene. And if you have actual curly hair, then you are probably homozygous for that particular trait. We've already talked about codominance in terms of the ABO blood grouping. So in particular, it's really just that AB group that matters, because A and B are completely dominant over O, but when they get put together, they're codominant. Another example of this is the roan pattern that we see in cattle and some horses, where if you are homozygous for the red, you have that sort of reddish brown coat color. Homozygous for the white will give you that albino white coloration. But if you have both one of each copies, you end up with this roan pattern where you're kind of red and kind of white. And you can see codominant, right? They don't merge together to make like a pink cow. They don't. They make a cow that's partly white and partly red. All right, so there's codominance. And then when we talk about the ABL blood grouping, this is another great example of multiple alleles, because you have different interactions. There are dominance hierarchies in this. A and B are completely dominant versus O, whereas A and B are codominant with each other. We also talked about this regarding the rabbits as well. Wild type is dominant over all others. So the C+, big C+, dominates over others. Chinchilla is recessive to wild type. So if you have the chinchilla allele, little c, superscript CH, chinchilla is a black tip white fur. It's an interesting coloration. If you have one brown-eyed allele, that rabbit's just brown. But if you end up in a situation where you merge it with a Himalayan allele or albino allele, you actually end up getting incomplete dominance. You get an intermediate phenotype. Same thing happens with the Himalayan, except with the Himalayan, it is dominant to albino. So what I'm trying to show you here is like, it's not even as simple as like combine this and you get that weird phenotype. Sometimes you get like complete dominance depending on what allele is involved. Sometimes you get incomplete dominance depending on what alleles are involved. And if a whole other gene allele shows up, it doesn't matter what the interaction with the other one is because it's completely dominant over it. So when you have multiple alleles, it gets very confusing for things like this. So just keep that in mind. Now let's talk about sex-linked traits. So sex-linked traits are inherited differently in sexually reproducing species than most other traits. This comes down to the fact that sex chromosomes can be different. All right. And the big issue here is because in many species, you end up with the male of that species having a different set of chromosomes than the female of that particular species. So we'll keep it fairly simple here. And we'll look at it in the context of species that have an XY sex chromosome pairing. There are other systems out there for determining sex. There are actually lots of different systems out there for determining sex, but let's stick with XY because it's just the easiest to understand. Okay. In humans, all right, and many other species, genetic males have a single X chromosome and a single Y chromosome. And then the females of those species are homozygous for the X chromosome. So first off, let's talk a little bit here about the terminology here. In this case, females are homozygous for the X chromosome. They have 2 copies of the X chromosome. On the other hand, males are going to be hemizygous for any trait on the X chromosome. So XX females can be homozygous for X-linked traits. They could be heterozygous for X-linked traits because they have 2 copies of each. On the other hand, males can only be hemizygous for X-linked traits. Hemi meaning half, because they only have 1 copy of the genes that are located on the X chromosome, because the other half of that chromosome pair is this runty little Y chromosome that doesn't match up at all with things that are on the X chromosome. And that's why inheritance now begins to depend on whether that recessive trait comes from the male parent or from the female parent. Because in most species with XY inheritance, well, in all species with XY inheritance, the way you end up with a genetic male offspring is the male has to provide the Y chromosome during the mating. So let's just look at this real quick. Male gametes, you can see here you have the X and the Y. Female gametes are XX. So resulting female progeny will end up having whatever's on dad's chromosome X and whatever's on mom's X chromosome. They can be theoretically homozygous or heterozygous for things. But over here, we can see that in the case of male offspring, the X chromosome has to come from mom because dad's only giving a Y that doesn't have that genetic information encoded from the X chromosome. So what we're looking at here is something in being inherited in fruit flies called the white allele. So XW. Okay, it's an X-linked trait. It's the eye color gene that's located on the X chromosome. And in the cases of this, you have a red-eyed male and a white-eyed female. The white chromosome, the W, actually, the big W turns the eyes red. I know it's confusing. It's because it's described after the phenotype. When they mutated this gene, the eyes turned white, which means the normal function of that gene is to turn the eyes red. So you get this weird sort of backwards labeling of, we call the gene the white gene, even though the product of the gene turns the eyes red. Okay, you're going to get that when you study genetics. Sometimes people name things after the phenotype rather than what it's supposed to do. So anyways, in this case, you have a male who is hemizygous for the red-eyed gene at this location, and you have a female who is double recessive. See the little Y. It's hard to distinguish that, or the little W. It's hard to distinguish the little W from the big W. Well, whenever this mating results in a female offspring, those female offspring have red eyes. Why? Because it's getting the dominant W allele from dad in both cases, even though it's getting the recessive W allele from mom. Doesn't matter. One round of that, one copy of that dominant red allele, the white allele, sorry, is enough to turn the eyes red. But when we come down here to the male offspring, they all have white eyes. And the reason why is that Y chromosome is what dad's giving. That Y chromosome doesn't contain any information for that white gene. And the only other copy it gets, hemizygous, remember, comes from mom, who has the recessive trait. So you end up with this weird inheritance pattern. Instead of getting what you would expect, if this were a normal gene inherited through somatic chromosomes, you would expect this to be a heterozygote with red eyes, and then you'd have that 1 in 4 chance of double recessive. You don't see that here because dad can't rescue the phenotype because he's giving his Y chromosome, not his W. So you get a weird inheritance pattern. And there are lots of sex-linked traits in humans. These are obviously much more common in genetic males than they are in genetic females, although it's not impossible for a genetic female to have a lot of these things. So for example, red-green color blindness is usually inherited mostly by males. It's an X-linked trait. If mom gives her son the colorblind version of that particular gene, they will be colorblind because dad's Y chromosome can't rescue that. hemophilia types A and B, Duchenne muscular dystrophy, Becker's muscular dystrophy, X-linked ichthyosis, X-linked agammaglobulinemia. This is a particularly devastating immunological disease. So is SCID. So if you've ever heard of Bubble Boy, Bubble Boy is a boy that lived to be about 13. He had to live essentially in a hermetically sealed bubble for his entire life because he has no immune system. People born with SCID essentially have no immune capabilities. And then glucose 6-phosphate glucose 6-phosphate dehydrogenase deficiency. That's a tough one to say. G6PDH. There's a reason they abbreviate that. So all of these are classic examples of sex-linked traits in humans. And as such, they tend to be found in significantly higher at significantly higher rates in genetic males, simply because dad's Y chromosome can't rescue the affected chromosome that's coming from mom. So in general, females tend to be carriers of these while the males are most likely affected. Now, it's not impossible for a genetic female to inherit these, but one of the things that I would say is, well, their dad probably has this. So if the father is red-green colorblind, and then mom is carrying a red-green colorblind allele, well, if they have a daughter, there's a 50-50 chance that it gets dad's 1 in 4 chance that it gets dad's red-green colorblindness allele and mom's red-green colorblindness allele, and they end up homozygous for this. So it is possible for women to be colorblind and have hemophilia and all these. But in general, they tend to be carriers because they are able to be heterozygous for X alleles. Males can't because of the XY mismatch. Another form of inheritance that I'll touch on are polygenic traits. So polygenic traits are when the phenotype is controlled by more than one gene. The phenotype then depends on the cumulative alleles present for that particular gene. We tend to refer to these as being inherited along a spectrum. It's also known as continuous variation. It's also one of the reasons why the blending theory of inheritance stuck around for so long prior to Mendel's work, because a lot of genes do appear this way. Essentially, when you look at stuff like height or skin color, to some extent, eye color, we'll talk about in a second, body weight, these are all inherited along a spectrum, which means you tend to have a bell-shaped distribution of phenotypes. So, for example, if you have somebody who's very tall up here in the 6, somebody who's very short down here at the 0, when you combine them, you're most likely going to get somewhere in this middle spectrum. There'll be somebody who's sort of intermediate in height, but it's not impossible for them to get some combination of this as well. So a 6 plus a 0 is always going to end up being homozygous or heterozygous, right? Because this person obviously only is homozygous recessive at 3 different genes. In order to be that short, this person is homozygous dominant at 3 different genes to be that tall. But you get combinations therein, and that's why it's not always surprising if you have a tall dad and a short mom that, the child might be reasonably tall, because it depends on how many of these boxes are sort of ticked at this point. And this is why you get this bell-shaped curve for a lot of these traits. Eye color is a little bit different. We did used to think it was encoded by a single gene. So the gene that we particularly think about this is ACA2, so OCA2 is associated with melanin-producing cells. This is really what determines brown eye, blue eye. All right, but these other 3 genes, HERC2, TIR, SLC24A4, these actually control more of like the type of blue or the type of green or the type of hazel that you have, and that's why eye color does occur a bit over a spectrum. Like I said, there's at least 8 genes in here. ACA2 is the one that really has the most prominent role. That's when we think brown eye versus blue eyed, and then the other genes contribute to the type of blue eyes you have or the type of brown eyes you have. So there is a bit of a spectrum here, but there's also some discrete traits that lie within there. So it's a little tricky to figure that out. But science is messy. Inheritance is messy. Genetics isn't as clean as you'd like it to be. That being said, genetics does come down to being one of the more mathematical, at least inheritance comes down to being one of the more mathematical parts of the biological sciences, because it really does come down to probability. So again, I have talked about this in some of my other videos. I'll show you a Punnett square here in just a minute. But in reality, what we're doing with Punnett squares is just making a graphical depiction of the probability of inheriting certain alleles from your parents. When we talk about the product rule versus the sum rule, it depends on what question we're asking. So, for example, if we're asking, what is the probability that the progeny, the offspring, will inherit a dominant allele from both parents, right? So, we want both things to happen. This is what we would call our and situation. I want to know what will happen if I get this and that, right? With the product rule. So, for example, let's say we're in a situation with complete dominance. We have a big B allele, a little B allele. Both parents are heterozygote, right? Both parents are heterozygous for this. Well, that means there's a 1 in 2 chance of getting big B from dad. There's a 1 in 2 chance of getting big B from mom. How do we figure out what the probability of inheriting both of those would be, i.e., having a child that is homozygous dominant, homozygous big B? Well, it's 1/2 times 1/2, and that is 1/4. And if you do your Punnett square for big B, little b crossed with big B, little b, you'll see in the upper left corner, that's where your big B is going to little b. There's a 1 in 4 chance of having that. Now, let's ask something different. What is the probability that the progeny will inherit only one allele, at least one allele from one of the parents, right? I just want one big B. That's it. That's all I care about. One big B. Well, that is what we would call an or concept. I want this or that. I get a big B from mom or from dad. That becomes the sum rule. So as we just described, there's a 1 in 4 chance of getting the big B from just mom. There's a 1 in 4 chance of getting the big B from just dad. But we can add those together because we're willing to take a big B from either one. We just want to have the dominant phenotype here. Well, that makes it 2 out of 4 in that particular scenario. So if you want both, 1 in 4. If you're happy with getting it from either, and one's good enough, 2 out of 4 using the sum rule. So like I said, Punnett squares are really just a way of doing math. So this is what we would call a dihybrid cross. The cross would be between two individuals who are heterozygous for 2 different genes. So in this case, you have the R gene, which is responsible for determining whether the P's will be round or wrinkled, with the big R round phenotype being dominant and the little r wrinkled being recessive. And then you have the Y gene, where the dominant phenotype actually is to have yellow P's, big Y, and the recessive phenotype is to have green P's, which are little y. So how do we figure this out? Well, it's simple. Across the top, we take one parent. What are the possible combinations that parent could pass down? Well, it could give a big R and a big Y. It could give a big R and a little y. It could give a little r and a big Y, or it could give a little r and a little y. All those are reflected here. The vertical axis is identical to the horizontal in this case because both parents have the exact same genotype. And then we just start moving things down and across. Big R, big Y, big Y. Big R, big Y, little y. See how they're all written out? We go all the way across. Now, all these are literally the product and the sum rules just being reflected graphically by doing this particular Punnett square. And in the end, we can go through and we can look, and what you'll end up when you have a dihybrid cross for 2 individuals who are heterozygous at 2 different genes, you're going to end up with a 9 to 3 to 3 to 1 ratio, where you have 9 individuals, 9 out of 16, all right? 9 out of 16 individuals are going to have dominant phenotypes for both genes, round and yellow. Then you have 3 individuals right here, bing, 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 who are dominant for the R allele. So they're round, but they are recessive at the Y allele. So they're green, round and green. Same thing over here, except it's the inverse, right? Now we have wrinkled P's that are yellow. And then down here, 1 in 16, you have your homozygous recessive at both alleles, your 1 in 16 wrinkly green P. All right. All that is math. And the reason we need to understand math is because if we're going to start doing a trihybrid cross, it's going to get real messy. we're going to go and have a lot of, now we're up to 64 different squares to do all this. And it gets very tricky to find out how to show all these and that kind of stuff. So it's often much easier to analyze a tri-hybrid cross like this, where you kind of just use your product rules. So for example, you know that again, they've got yellow, you've got the yellow versus green, the round versus wrinkled, and the tall versus dwarf. So again, you've got yellow being a dominant allele, round being a dominant allele, tall being a dominant allele. And you know that it's going to be a 3-to-1 outcome, right? 3-to-1. And at the next level, 3-to-1. And at the next level, 3-to-1. So if you want to calculate the probability of any of these phenotypes being combined, you just go 3 times 3 times 3. You'll have 27 that are yellow, round, and tall. They have all 3 dominant phenotypes. 3 times 3 times 1, 9 of them will have the dominant yellow, dominant round, but the recessive dwarf phenotype. 3 times 1 times 3 gives you the 9 yellow, wrinkled, tall. And you see how you're just using these product rules to say, I want to have this and this and this. And you're just, what's the probability of each in each case? Doing it with math is going to be significantly faster in these cases. And then again, if you need to do 4 different genes, then you add a whole other row and it just keeps going like this, which is much easier than drawing 128 square Punnett cross to do your 4-factor cross at this point. So what I'm getting down to it is I know a lot of my biology friends, we really don't enjoy doing math. There's a reason we went biology instead of chemistry in some cases, or even physics for that matter. But it's fairly simple math, and it's going to save you a lot of time versus drawing one of these big old Punnett squares when you're trying to do one of these large-scale genetic crosses involving multiple genes with multiple alleles. So I hope this video gives you a little more clarity on the different types of inheritance, how we can use mathematics and Punnett squares to help learn information about how crosses should come out, and using that information from doing crosses to figure out what type of dominance we're looking at. Is it complete dominance? Of course, here we're looking at situations where all 3 of these are completely dominant alleles. This is going to get even more confusing if we start throwing in things like incomplete dominance, epistasis, and all these other things that I've talked about in other videos. So I hope you enjoyed it. I hope you learned a lot about the different types of inheritance and how this stuff works. And I hope to see you again later. Talk to you soon. Bye.